Over the last few decades, CO2 emissions have been at an all-time high due to human activities. This has been causing drastic alterations to the environment, threatening not only humans but marine ecosystems as well. Global warming is suggested to play a major modulatory role in the occurrence and geographical distribution of PKD in species related to salmon. The disease occurrence may be increasing in areas that have historically been cold enough to prevent clinical outbreaks. With the rising temperature of oceans due to climate change, this has proliferated the number of parasites in bodies of water and given an advantage to their life cycle, as the increase in temperature is more suitable for the parasite's life cycle and suppresses the salmonoid immune response.
This led us to ask the question, with the increase in climate change, how does this affect the proliferation of the parasite tetracapsuloid bryosalmonae, and what does this mean for the populations of salmonid species? The increase in water temperature that was stated earlier is suspected to be the reason for the proliferation of parasites. Scientists assume that as the temperature of the water continues to rise, this allows for an increase in the secretion of the infectious spores by the bryozoan host. In the research article by Fontes et al., it stated, temperature also had an effect on the presence of T. bryosalmonae in the river water, with a unit increase in temperature increasing the likelihood of presence by 1.6 times. In this same study, they had tested various rivers, River Avon, River Dun, and River Itchen. River Avon was recorded to have the highest temperature as well as the highest detection of spores versus River Itchen, which was recorded to have the lowest temperatures and also had the lowest spore count. This gives us further evidence into the effect of global warming on the increase of T. bryosalmonae in our waters.
The parasite Tetracapsuloides bryosalmonae is a myxozoan, or an endoparasitic animal. They are part of the Cnidarian phylum. The parasite's life cycle is complex and entails utilizing vertebrate and invertebrate hosts. For Tetracapsuloides bryosalmonae, their hosts are most commonly the freshwater bryozoans and various fish in the salmonid species. The parasite starts off as a single cell described as a covert stage or avirulent stage that shows no effect on the bryozoan's normal function. As the parasite becomes multicellular, this is termed the overt or virulent stage, reducing growth and increasing mortality of the host. Infectious spores created in the body cavity are released through the pores at the base of the host. These spores target and enter into the salmonids and make their way to the posterior kidney. It is here where the cells begin to multiply until they create a mature sporogonic pseudoplasmodium that is released through the urine and is viable to infect the freshwater bryozoans, completing the life cycle. We mentioned earlier that the outbreak of the parasites has been linked to the increase in temperature. According to the study conducted by Bailey et al., quote, at lower temperatures, fish exhibited an asymptomatic version of the disease and lower parasitic burden. This can be contrasted with warmer temperatures. Quote, the increase in only 3% appeared to impact the parasite proliferation in the biological and biochemical processes of the host, with the lower temperature being less detrimental to the host's immunocompetence. This shows that the parasite is able to infect the salmonid easier due to the host's weakened immune system for the rise in temperature, strengthening the overall life cycle of the parasite.
The infectious spore invades the salmonid host by way of their gills and migrates through their circulatory system till it gets to the posterior kidney. Once the parasite has landed in the kidneys, Bartolomeo Gorguilon et al. Stated that it is triggering an aberrant histozoic extrasporogonic proliferation that causes a chronic immunopathology with host immunosuppression, often predisposing the fish host to secondary infections. The most common secondary infection seen is proliferative kidney disease, PKD. PKD can cause a list of symptoms in these fish, including nephromegaly, splenomegaly, pale gills, darkened skin, etc. As we stated, global warming also plays a role in PKD seen in salmonid species. As the temperature of the water increases above 15 degrees Celsius, this provides the environment to cause secondary infections such as PKD seen in the salmonid species.
The parasite is able to thrive in different areas due to the rise in temperature of the bodies of water across the globe, where otherwise these parasites may not have been able to maintain their life cycle for as long in quote normal conditions. In colder areas, we may not have seen this parasite live, where we are now seeing them proliferate. In this graphic, we can see the vast range of species across the world that are being affected by this parasite.
PKD seed and salmonid species is economically important in the related industries located in North America and Europe. Wild brown and rainbow trout in some rivers in Switzerland have populations that are infected from 40 to 100%. Although wild species are more likely to be infected, it is still possible for the farmed salmonid species to become infected as well. In fact, the numbers of fish infected for each group, wild and farmed, are growing annually. Bartolomeo Gargiulo et al. Stated, recently T. bryosalmonae and record-breaking summer temperatures contributed to a massive fish kill of thousands of mountain whitefish in 2016 in the Yellowstone River and its tributaries, with a drastic impact on the recreational fisheries and economics of Montana, USA. This impact from the parasite infection and secondary diseases is reaching across the northern globe and has touched the fishing economy in a negative way. While investigating this parasite, we realized that there was limited information that exists on T. bryosalmonae in North America when compared to Europe. I think that researching more into the strain of parasites here in North America could give better insight into how our strain is affected by the environment and what we can do to help suppress it from destroying our fishing economy. We also were wondering as well, how will ongoing climate change further proliferate the amount of parasites present in the ocean, and what will this mean for the marine ecosystems in long term?